Hello and welcome to the next video in the A-Level Biology series. In this video we will finish our look at inherited change. In this video we will cover gene mutations, genetic disease, and the control of gene expression in prokaryotic and eukaryotic cells. Genes encode proteins. An abnormal gene can lead to the synthesis of an abnormal protein which can affect the phenotype of an individual. DNA undergoes transcription to form mRNA, which goes on to be translated into a sequence of amino acids or a polypeptide, and then folds to form proteins which give rise to the phenotype. For example, blue eye colour. A change in the sequence of base pairs in a DNA sequence may result in an altered polypeptide, affecting the structure and/or function of the protein the gene encodes. This is known as gene mutation. Mutations can lead to misfunctioning or non-functioning proteins, causing disease. There are three types of gene mutation. One is insertion. This is when a nucleotide with a new base is randomly inserted into the DNA sequence. This can change the amino acid encoded in the triplet or cause a frame shift. A frame shift is when nucleotides are added which are not divisible by 3. Due to the reading of DNA working by triplets of bases, this causes a shift in the reading frame which can result in a totally different translation. 2 is deletion. A nucleotide and base or number of nucleotides can be randomly deleted from a sequence, leading to a change in the amino acid sequence or a frame shift. 3. Substitution. A base in the DNA sequence is swapped for another. There are 3 different types of substitution mutation. Silent. This does not change the amino acid which is encoded. Missense. This alters an amino acid to another. And nonsense. This mutation creates a premature stop codon, making the polypeptide incomplete, thus affecting the structure and function of the resulting protein. Here are some examples of diseases caused by abnormal genes. Huntington's disease. This is caused by mutations in the HTT gene, which encodes for Huntington. This gene is found on chromosome 4. The disease affects neuronal development, leading to neuronal degeneration. Patients gradually lose ability to talk, walk, and think over time. The genotype is homozygous or heterozygous, as the gene is dominant. Sickle cell disease, which we covered in the previous video, is caused by mutations in the HBB gene. This is found on chromosome 11 and affects the structure and function of hemoglobin. The abnormal gene results in abnormal structure and function of hemoglobin. Blood cells form a sickle shape, unable to carry oxygen, and cells stick together, forming blockages. A heterozygous genotype means you have sickle cell trait. A homozygous genotype means you have sickle cell disease. Albinism, which is caused by the gene TYR, which encodes melanin and is also found on chromosome 11. The TYR gene encodes an enzyme involved in melanin production. This results in loss of pigment in hair and skin. The gene is recessive, so a person needs to be homozygous for the abnormal allele. The nucleus of every cell in the body contains the same genes. However, not every gene is expressed in every cell. The expression of genes is a tightly regulated process. let's discuss 4 important types of genes and enzymes which work together to maintain this control. Regulatory genes. These code for a protein which help to control the expression of another gene. Structural genes code for a protein which has function within the cell. For example, factor VIII, which is involved in blood clotting. Inducible enzymes. These are only expressed when their substrate is present. Repressible enzymes are synthesized as normal until a repressor protein binds to an operator on a gene. These are essential mechanisms in cells as it prevents the waste of materials and energy on low priority functions. Key regions of genes are an operator, a genetic sequence which allows proteins responsible for transcription to attach to the DNA sequence. A promoter is a sequence of DNA needed to turn a gene on or off. The process of transcription is always initiated at the promoter. A good example of gene control is the lac operon in bacterial cells. The lac operon in many bacterial cells is required for the transport and metabolism of lactose in the cell. This is the structure of the lac operon. First is the promoter for the regulatory gene, which is LAC-I, which encodes the lac repressor gene. Then there's a promoter for the structural genes, which is followed by an operator, LAC-Z, LAC-Y, and LAC-A. LAC-Z encodes for lactase or beta-galactosidase enzyme. This breaks down lactose into glucose and galactose for energy in the cell. Lactase is a repressible enzyme, so it is only expressed when its substrate, lactose, is present. LAC-Y encodes for a permease enzyme, which allows more lactose into the cell. LAC-A encodes a trans-acetylase enzyme. It transfers an acetyl group from acetyl-CoA to lactose. So this diagram shows what happens when lactose is absent. When lactose is absent, the cell does not require the enzymes needed for transport and metabolism. LAC-I is expressed producing the lac repressor protein. The lac repressor protein contains 2 binding sites, one for lactose and the other binds to the operator in the lac operon. In the absence of lactose, the lac repressor protein binds to the operator region in the lac operon. This blocks RNA polymerase binding to the DNA and transcribing the structural genes. When lactose is present in the cell, the structural genes need to be expressed to transport and metabolize the lactose for use in the cell. Lactose binds to the binding site on the lac repressor, so it can no longer bind to the operator. RNA polymerase can now bind to the promoter region of the lac operon. The mRNA is produced with the transcription of structural genes, which can then be translated into the proteins. The enzyme lactase is produced, and now the cells can metabolize lactose in the cell. In human cells, or eukaryotes, proteins called transcription factors are used to control gene expression. Approximately 10% of genes encode transcription factors. Some transcription factors bind to DNA in the promoter region of the gene, resulting in activation or repression of gene transcription. An example is a PIF transcription factor in plants, which activates the expression of the amylase gene. Gibberellin is a plant hormone which controls seed germination by stimulating the synthesis of amylase enzyme. Amylase hydrolyzes the starch store in the seeds into metabolizable sugars for use as the cells grow and the seed germinates. Like the activation of the lac operon, a repressor protein will prevent the expression of the correct genes. DELA is a repressor protein in this scenario. It must be broken down by gibberellin to synthesize amylase. The following components are involved: the amylase gene and promoter, gibberellin, a gibberellin receptor and enzyme, and the DELA repressor protein. DELA protein binds to the PIF transcription factor, inhibiting it from binding to the promoter, so amylase is not expressed. Gibberellin binds to a receptor and enzyme complex, which activates it to break down DELA protein. Now the DELA protein is broken down, PIF can bind to the promoter region, activating the expression of genes and synthesizing the amylase protein. Now to summarize. Gene mutations can be caused by insertion, deletion, or substitution of nucleotides into the DNA sequence, which may cause the synthesis of abnormal proteins. Gene expression is controlled by regulatory and structural genes. Prokaryotes control gene expression from an operon, a cluster of related genes controlled by a single promoter. Eukaryotes control gene expression by regulatory and structural genes and the activity of transcription factors binding to gene promoters. Gene control is essential for reducing waste of resources and energy in the cell. This concludes today's video on inheritance and genetics. Thank you for watching today's video. We hope to see you next week for the next video on selection and evolution.